Okay, so I want to go over this, and I want to make sure everybody has some help. I want you to spend some time today uh, doing it in class. I have to miss today because of that water heater issue. I have to deal with it. Unfortunately, I have no choice. But of course, that'll mean that all the due dates are going to be delayed by a day, just so that we can have time together in class before the final submission is completed. Let's go down our objectives here. We're going to use the key terms to help understand and complete these objectives. These key terms, and I think you'll note that most of them we've used in class, but we'll look at some of the ones we haven't here in this video. We're going to discuss reasons for evolution of sex in organisms, and we've already answered this in class, and you should be able to think of why would sex be an advantage, and hence why would it evolve? Because remember, as we discussed earlier in the year, organisms were single-celled at one point and did binary fission, which is not sex. So what allowed for sex to evolve? What would give sexual creatures an advantage over asexual creatures? Asexual creatures would mean creatures that reproduce without sex. Know that meiosis, like mitosis, involves DNA replication during interphase of the parent cell, but that it is followed by 2 cycles of nuclear division. This statement implies, and I think it pretty much clearly states, that there is no DNA replication between the 2 cycles, and I've mentioned that, and we'll go over that again together. Recall how chromosome numbers can vary between somatic cells. Those are those cells that are 2N. Somatic means body cells. So somatic means body cells. That's every cell in your body except for your gametes, right? In gametes, in humans, it's sperm and egg, but it's those cells that only have 1 copy of each chromosome, 1 copy of each gene. In more detail, then number 3, summarize the principal events in meiosis. All right, so you saw here we kind of went over a real quick, number 3, we went over a quick kind of outline of what meiosis is. So what are the major steps of meiosis? We're going to summarize it in synapsis, and we've talked about that in class. Chiasmata. All right, that's the place of crossing over. The chiasmata is where the crossing over occurs. Separating the chromatids, second division, and identifying the names of the main stages. So you should be able to do all 4 of these. I think you should be able to do them by now. So if you're not able to do it by now, I'd like to know why. So when I see you on Thursday morning, if you wouldn't mind dropping by, or after school on Thursday, why don't we discuss why is it that you don't know these 4 steps, since we've gone over it. And I think you'll see if you look at the video that I'll send you that we covered in the material we covered in class. You'll see we did cover this in class, and everyone seemed to understand it. So 5 is pretty much done. Describe the behavior of homologs and their associated alleles during meiosis and fertilization with reference to independent assortment, paternal chromosomes, recombination segments of material, and parental homologous chromosomes in crossing over. We'll talk about the details of these steps as we go through this video. The random fusion of gametes during fertilization. Explain how these events create new allele combinations in gametes. So we'll discuss that. We've discussed it already. Again, you can refer back to the previous video, the class video, not previous, because I haven't sent it yet. Explain the significance of linkage and recombination to the inheritance of alleles. We'll discuss what linkage is and why it's important. Explain what is meant by the crossover frequency. Demonstrate use crossover values in chromosome gene mapping. Again, we're not going to focus on gene mapping, and in fact, if we don't do any gene mapping, we're fine. We can do some of that if you'd like after the end of course exam, but I like to focus on more relevant points, only because we're behind the 8 ball with time. So you should understand some chromosome structures, points like what are histone proteins, those things that you wrap DNA around. You should know what those are. I would guess that by this point that you would know that you would start. I said you could use this as a study guide, so you should know these are your vocabulary words, your key terms. You should know what those mean, right? You should know what each of these steps are, because I'm telling you they're important, but also because I told you this was a study guide, so there's a lot of information there for you to use to identify what's important from the chapter. So you should be able to tell the difference between prokaryotic and eukaryotic chromosomes at this point. At least some simple distinguishing points. For instance, that eukaryotic chromosomes are linear and prokaryotic chromosomes are circular, right? That seems to be a simple one. Define the terms karyotype, autosome, sex chromosome, explain karyotyping. You should be able to do that. There's a whole little paper lab they should have done by now. It's due Thursday, so I'd assume that you completed it by now. Some of the kids were getting help yesterday after school, or actually I guess it'd be today after school. It's night, or it is yesterday because it's 12:30 right now. So no, karyotyping uses cells collected by chorionic villus sampling and amniocentesis for parental diagnosis of chromosome abnormalities. We may or may not talk about this. It's important, but again, we have to, because of time, we have to consider whether this is going to be something we want to focus on. Amniocentesis is the way we can collect chromosomal data on a fetus while it's still in the womb. It's an interesting process. We'll see if we can get to it, but we'll try. So let's start with meiosis then. General outline. There's nothing on this page that's not covered in the classroom. It hasn't been covered in the classroom. It has been covered in the chapter as well. But I just think there's some really important steps that I want to point out here. And as you're looking at your paper, maybe you should highlight these as well. And I've discussed them in class and highlighted them in class as well. First of all, if you get a mutation here during meiosis, what's interesting is that the mutation happens in the gametes. If there's changes in the DNA at this stage, then every cell of your offspring will have those changes. So that's really interesting, because let's say there's a skin cell on your hand and you're outside playing basketball all day and it's really sunny, and that sun exposure, that sun overexposure causes a genetic change in a single cell on the back of your hand. Well, that's not going to change all the DNA in every cell of your body. However, if you change the DNA in your gametes, which would be in your testicles or your ovaries, if the DNA changes there and you have children, then your children will carry those mutations, those mistakes forward. Now remember, those changes, mistakes, if you will, those changes or mistakes, and we've covered what kinds of mutations, what are mutations and the different types. If those occur here in the gametes, then those genes will be in every single cell of your child's body. So it's really interesting that meiosis is producing the gametes, the very first cells of a new living organism, or at least half of the first cell of a living organism, right? This is what chromosomes in anaphase of meiosis look like. Again, it's you'll see under the microscope when we do the lab, but this is kind of condensed. They're stained, or you wouldn't be able to see them very well, but they're condensed. They're wrapped around histones. Actually, this isn't a stain. This is an electron micrograph picture, it looks like. All right, so just kind of a picture of what they look like. These are drawings, obviously. All right, so this in interphase, you're not dividing this. You're not dividing yet, right? If this was not a dividing, if this cell was not dividing, we wouldn't see these beautiful little sticks, these chromosomes. We would just be, all we would see is just a bunch of unraveled spaghetti, black, dark center. Meiosis is, before you even start meiosis, remember begins here at prophase 1, you have to duplicate the DNA and you move through it, you make copies, so instead of having one little stick, right, you have two, and you can see them there, there's two blue ones and two white ones here, there'd be two of these and two of these. And then you have two, and so there's two chromosomes, chromosome number 1s, and two chromosome number 2s. In this example, this one came from mother, this one from father, or vice versa. So these two came from mom, these two came from dad. So during DNA replication, we make copies of them. You remember semi-conservative replication. They kind of stick together using at the here at the centromere, they stick together. And we will look more on chromosomal structure in a second. So there you have, they're duplicated. In prophase 1, they condense. They look more like these do here. And synapsis occurs. In other words, the two homologs, we call them homologs, the two chromosome number 1s are homologs. Remember, the sister chromatids are identical. The homologs are not identical. They're similar. They have the same genes, but they'll have different alleles, right? We'll talk more about alleles in a second. So when you have a tetrad or bivalent means two, bi means two, bivalent or two chromosome structures. Tetrad because there's four chromatids, bivalent because there's two chromosomes, right? There's crossing over here. Crossing over occurs, and you switch some DNA. That all happens in prophase 1. Also, you'll note that the nuclear membrane starts to dissolve away. The centrioles, you didn't have them in interphase, now you have them. And the spindle fibers start to form. Again, all this is covered in the previous video, but I just want to make sure that you see it here in the drawing. By the time they start lining up, what you'll see is in metaphase 1, the centrioles are on each side. The spindle fibers are attached at the centromeres, and now you're going to separate not the individual chromatids the way you did in mitosis, right? Remember, in mitosis, we separated the two chromatids. Here, what you're going to do is separate the homologs. So you're going to separate the two chromosome number 1s and the two chromosome number 2s. What's interesting, and there's some really subtle differences, but they're important differences, is in mitosis you'd have one chromosome from mom here, one chromosome from dad here, one chromosome from mom here, one chromosome from dad here. In this, when we separate the homologs, it's going to be random. So what's going to happen is that you're going to separate the two chromosomes, but you'll note that chromosome number 1, you only got it from dad. Mom's chromosome number 1 is over here. And over here, chromosome 2 is from mom, and chromosome 1 from dad's over here. So what you see is that there's a random separation of the DNA. Already you have a difference in DNA between these two. In mitosis, because you separate the sister chromatids, which are identical, the two cells have identical DNA. But because in meiosis you separated the two homologs, Now you have just without even discussing crossing over, you've already made a difference. If mom had the recessive trait for a specific gene on the chromosome number 1, and dad had the dominant gene, then now you've separated the one cell has the recessive gene, the other cell has the dominant gene. Now, what's interesting is that these sections of the DNA crossed over, and here the crossing over happened in the middle part of right in the middle of the one arm of this sister chromatid that's hanging out. You see the blue and the white, they exchange DNA, and so now you have a little bit of moms over here on dads, and you have a little bit of dads on mom's sister chromatid. And that's that other important piece, and that important piece is that you're recombining the DNA on this sister chromatid, right? Where you used to have only the dominant gene here from mom, now you have the dominant gene still on this sister chromatid that it didn't cross over, but the recessive gene is now here. And dad, where you just had the recessive gene from dad, you were heterozygous for this trait. Now you have mom's recessive gene with that, or dominant gene with that. So moving forward, we'll look more into crossing over in a little bit. You'll see that we have two cells. We started off with how many chromosomes in each? We had 2 number 1s and 2 number 2s, so that's 4 chromosomes in total. Here, what do you have? You have 2 number 1s, in other words, 2 big ones. They're sister chromatids, but if we separated them right now, if they separated automatically all by themselves, then you'd have 2 number 1 chromosomes and 2 number 2 chromosomes. But because they're connected by a centromere, we don't call them chromosomes, we call them sister chromatids. So there they are. We have only 2. We had 4, now we have 2. So we've actually already technically gone down to one end, from 2 end to 1 end. But we do have still 2 copies, right, of each chromosome here. So it depends how you look at it. We need to separate these 2 copies. So how do we do that? We do it by doing this whole process again, but notice we're not making a copy of the DNA. So we're skipping this step where we went from 1 to 2 copies each. We're skipping that step, and we're going to divide these. Now this looks a lot more like mitosis, where the 2 sister chromatids move to opposite sides of the cell. They're no longer identical, though, because they've switched DNA. You see that here, right? Crossing over did occur there. And so these 2 chromosomes are not identical. That's the ones we're kind of following and making sure we understand. So then they're separated now into 4 cells. Now in mitosis we made 2 identical cells. In meiosis you're making 4 not identical cells, and you've halved the DNA. You went from 4 chromosomes to 2 chromosomes in each of these gametes, an egg or a sperm. As you did in the lab, that was, again, it was going to be due today, but we'll have to move it back. And there's a really great example by one of your colleagues that was done. Hopefully you do your own copy. You don't copy what she did. It was fantastic work, and yet hopefully you see that there's a real important reason why we went from 2 N up here to single N in each one of these cells. All right, let's go on. So that's the way meiosis is supposed to work. But sometimes we have something called nondisjunction. And what that means is this: instead of the chromosomes separating the way they should, by the way, disjunction. What does the word junction mean? It means a place where they come together, right? Disjunction is if they're coming together at the junction, disjunction must mean they're coming apart. Non-disjunction means that they're not coming apart. It's kind of crazy. I'm sure some evil genius English teacher or science English/English major decided to write that, but whatever. Non-disjunction just simply means that there's a failure to separate the chromosomes. So here you're coming up, and you're supposed to have one of the chromosome number 1 pair of sister chromatids on one end and one on the other. But in meiosis 1, when there's non-disjunction, what we get is there is no chromosome number 1 here. And there's 2 copies of chromosome number 1 over here. Why? Because they did not separate the tetrad or bivalent chromosomes. The 2 homologs didn't separate correctly. The junction was not, they weren't separated. They stayed together. So what you get when you separate them again in meiosis 2, okay, when you separate in meiosis 2, so meiosis 1 we separated the homologs, these didn't separate, so now you ended up with an extra pair of homologs here. With meiosis 2 we separate the sister chromatids, and what you end up with is what? 2 chromosome number 1s and only 1 chromosome number 2 in each of these new cells, right? In these 2 cells, you only have chromosome number 2. So that's a problem. You're going to have too few chromosomes over here and too many chromosomes over here. When you have an abnormal, which means, and this is important because most of these gametes end up in some type of miscarriage because they're not really going to be able to function as living organisms. Abnormal chromosome numbers, we call that abnormal chromosome number an aneuploidy. I want to point out that some non-disjunctions do end up giving you an aneuploidy or abnormal chromosome number where the individual is able to live a normal lifespan. But many of them, unfortunately, or fortunately, depending how you look at it, are not able to successfully complete their development. So non-disjunction in meiosis 2, though, let's say it happens in meiosis 2 instead of meiosis 1. So here we're able to separate the homologs fine, but then when we go to separate the sister chromatids, this pair of sister chromatids don't separate. The junction stays. There is no disjunction, so non-disjunction. There's no separation. So you end up with only 1 chromosome number 1 here, no number 2s, and 2 chromosome number 2s, and only 1. Where over here, you have this normal number. Now you'll notice if it happens in meiosis 1, then all 4 have an aneuploidy. They're all 4 gametes have either too few or too many chromosomes. Where if it happens in meiosis 2, then you have the 50-50 shot, 2 out of 4, having a normal chromosome number, and 2 of them having an abnormal chromosome number. So I pretty much did number 1 already for you in this little explanation. And I did number 2 for you. If you rewind and listen to it, you can put in your own words. Explain how mitosis conserves chromosome number while meiosis reduces the number. I think I've explained it a lot already. But remember mitosis, how is it that we do it? We duplicate it and we separate the sister chromatids without ever separating the homologs. I mean, I can't say it in many more different ways. Go back and rewind this video. You can listen to or we'll go over what we covered in class or read the textbook. Both these light micrographs, A and B, show chromosomes in metaphase of meiosis. So they're both showing meiosis and metaphase. Notice how the chromosomes are all lined up on a plate. Aren't they beautiful? How are they different? What's going on here? So they're both metaphase of meiosis. What's the difference? Look at these two and describe the difference. You tell me. By the way, see how they're lining up? I think this is clear as you can be, and this is an actual picture of metaphase. Let's move on. All right. So when we're looking at crossing over, what are we talking about? And again, we covered this in class. I don't want to spend too much more time on this. There's 52 pages to go through. and it's already late for me. So a centromere, that's where the two sister chromatids are connected to one another. After you make a copy of the during DNA replication, they're stuck at this. They're joined together at this place called a centromere. They're exact copies, so this DNA is the same as this DNA here, right? So we call them sister chromatids. These two are sister chromatids. These two are not sister chromatids, right? This came from dad and this came from mom, or vice versa, whatever. But notice that the two homologs, we call these two homologs, there they are, homologous pairs or homologs, right? Notice that they have the same genes. These all have gene A, these all have gene B, these all have gene C, and these all have gene D. But in this particular case, and I believe in the example I did in class, what you'll see is that, dad was, let's say Blue is dad, dad's chromosome genes were both dominant, and mom's were both recessive. But they controlled the same protein. They did the same function. It's just this had a version that doesn't show up when you have this version in the same cell. So this person who's doing this meiosis, from their father, they received two dominant alleles on these two genes, and from their mother, they received two recessive alleles on these two separate genes. Remembering that, they say maternal chromosome right here, and this is the paternal, so this is father's and this is mom's. So dad is giving the recessive and mom the dominant. I didn't notice that. All right. Okay, so moving forward, synapsis occurs. Synapsis allows the homologs non-sister chromatids to become entangled, and the chromosomes exchange segments. So you're going to get an exchange of DNA. Notice what's happening. They get entangled or wrapped around each other. This section of DNA is moving over here and joining with this section, and this gray section is going to move over to this blue section. And so what you get is a sister chromatid down here that's no longer identical to this sister chromatid. This sister chromatid is no longer identical to this sister chromatid. So up here you had 4 chromatids, 2 50-50, 2 different types, all the recessives or all the dominants. Down here you have 4 different types, right? Yeah, you've recombined the DNA. You've rearranged the DNA. Recombined is the same as rearranged. You recombine the DNA. Now you had, instead of having all capital or all dominant forms of the gene, now you have all dominant forms on this sister chromatid, except for D, where you have the recessive form. Of course, there could be hundreds of genes on this section, of the chromosome, but in this case, in this example, we're looking at one gene. And instead of having all those recessive versions of all 4 genes on this chromatid, it does on this chromatid rather, on this chromatid, they used to be an exact copy, now has all recessive except for one, which is as the dominant. So we've recombined them, or we've mixed them up. That is what recombination means. This exchange, this place where they're exchanging, this crossing over event, is happening at the chiasmata. That would be the plural, right? The singular version is chiasma. So this is where it's happening, the chiasma. All right. So once we're done making copy or completing the final division of meiosis, the 2 sister chromatids that made up each replicated chromosomes become separated and now referred to as chromosomes. So these 2, once they're separated, now will become chromosomes, and I've said that before numerous times. So expect that might be tested. 4 genetically different chromosomes are produced. So here you have 4. Instead of having just 2 different kinds of chromosomes, now you have 4 different kinds of chromosomes. Recessive and dominants are mixed up. Now you should be able to answer these 2 questions pretty easily. This is a really nice comparison between mitosis and meiosis. Notice that the mitosis, they're all lining up. There's still you replicate the DNA, so there's 2 copies on each chromosome, hence the sister chromatids, right? Those are the copies. They line up on the metaphase plate. You saw a light micrograph picture, a light microscope picture of this. But the homolog chromosomes do not pair up the way you're going to see them do over here. They don't do synapses here. And so when they line up, they get the sister chromatids get separated. One pair of sister chromatids going this way, one the other. One of each, so that this DNA is going to be identical to these 2 DNAs, etc., etc., etc. In mitosis, you went from 2N, 2 copies of each chromosome, not including the extra copies you made after duplication, right? Or replication. You go through mitosis and you end up with 2N. You started with 2N, you end up with 2N. They're identical genetically. There's no crossing over because there's no synapses. So mitosis is exact copies, barring mutations, which could happen anywhere along the process, right? So meiosis happens, it's doing mitosis twice. The first step of meiosis is reducing the number of chromosomes. So one of the first things we do. But also in the first division, you're getting crossing over happening. This is where you're starting to increase genetic variation. And the random assortment of the chromosomes, whether you get dads over here, moms over here, they don't organize their separation as well. This is all fairly random. So what you end up with is a greatly increased genetically varied cell, right? And you'll have 4 genetically different cells. So the first division separates the homologs, as you see here. The second division separates the sister chromatids. But because you have the DNA, when you separate the chromatids, you only have one chromosome in each cell. And you'll notice that each cell is different. This is a solidly, I think this is supposed to be maternal chromosome here. And here you have a maternal chromosome with some of the paternal or mother's chromosome with a little bit of father's DNA in it. And then here you have, again, pretty much the same thing happening over here. You have 2 cells over here where you have mostly dad's chromosome, but a little bit of mom's chromosome in there. All right. So each one of these is genetically distinct, and they're only 1N. There's only 1 copy of each chromosome. Here you have 2N, 2 copies of each chromosome. All right, you should be able then to explain all that pretty clearly. Zygote, by the way, zygote is what we call it when this sperm meets the egg and creates this new creature. We don't call it an embryo because it's not developed yet enough. We don't call it a baby because it's not developed yet. It's just a single cell. When you have that single cell, we call it a zygote. All right, so linkage is just simply when you have a linked group or linked pair of genes, you have genes that are on the same chromosome. Big A, big B, A and B, for instance, are both on the same chromosome. This is chromosome 1, let's say, and so these are chromosome 1 from this person's mother or father, it doesn't matter which, and this other chromosome is from the other parent. So 1 chromosome from this parent's parent, right? And this other chromosome is from this parent's other parent. So you'll notice that the genes A and B are found on the same chromosome. So the A and B are linked. Big A and big B are linked, and little a and little b are linked. Here again, little a, little b are linked, and little a, little b are linked here. So because they're linked, you have limited amount of. Again, we're not going to consider crossing over at this point. Let's just think about this. Because they're linked, because they're in the same group of, they're going to move together. So big A, big B are going to move together, and little a, little b are going to move together. They're going to get separated. So you're going to get big A, big B here, and after meiosis, and you're going to get, you have two combinations. You either get big A, big B, or you get little a, little b. Of course, there'll be two of these, but we don't have to show both, right? And you'll notice it says here that only one from each is shown. The only reason you need to show one is because these two are the same, right? This chromatid is the same as this; they're identical. So you have two two new gametes with. There would be four gametes, but they'd all two of them would have this this linked group, and two would have this linked group. In other words, big A, big B. The other one, little a, little b. This person's heterozygous, right, for both of these genes, big A and big B, little b. All right, this person here, this parent, was homozygous for both A and B, so little a and little a, little b, little b. And these two are linked, and these two are linked. In other words, they're on the same chromosome, so this is one link group, this is another link group. So again, you can form this many gametes, and the thing about linked groups is that restricts how many different kinds of gametes you can make, right? So instead of four different ones here or four different ones here, you have only two different ones here, and really you only have one kind of gamete you can make here, right? There it is, two recessives linked. There's only one kind. So now you can recombine. You're combining here. In other words, this is what happens in sex. The egg and the sperm come together, and so this let's say this is the father. Again, it doesn't matter, but this is the sperm and this is the egg. They come together. Then you get big A, little a, and big B, little b. So two heterozygous traits. If this one comes together with this one over here, then you get big A, big B, little a, little b. So again, two heterozygotes. If this one, however, comes together with this one, and you see the arrows here, this one and this one come together, then you have homozygous for A and homozygous recessive for B, so both of them are homozygous recessive. And then lastly, if this one combines with this one, again, two homozygous, so you get little a, little b, little b. So you only have really two different kinds of offspring you can have here, or two different kinds of zygotes you can produce. You can produce a heterozygous zygote or a homozygous recessive zygote. So only two options you have here. A far cry from the genetic diversity of two heterozygotes, right? Because why is this so different? Because these are homozygotes, right? All right. So what if these genes weren't linked? What would be the possible gametes? So first let's talk about the fact that these are linked means they move together as groups, and that limits the amount of the different types of gametes you can produce, the different combinations that you can make. So what does it look like if A and B were on different chromosomes? Well, I would draw it out if I were you. I'd draw it. Here's an A, right? And then some other chromosome in the same parent, right? Let's do first this parent, number one. They have big A and little a, right? So they have on this chromosome, they have A. On their other chromosome, of the same chromosome number one, let's say, they have little a. They're heterozygous for that trait. So they have little a. But on a separate chromosome, they have big B, and they have another chromosome, little b. And then that person marries another human with a recessive, two recessive A's, and then two recessive B's. So what kind of genetic variation, what does this do to genetic variation if these genes were not linked? In other words, they were on different chromosomes. What would that look like when we start to produce the gametes? Well, let's here put four gametes. Let's go ahead and put 4 gametes. Again, what am I doing? I'm just doing what they're suggesting, right? What it would happen if the genes were on different chromosomes. So I'm going to go ahead and do it. I'm going to go and create 1, 2, 3, and 4. 4 gametes there. I'm going to go 1, 2, 3, and 4. I'm going to do more than they did. I'm going to go ahead and draw all 4 of them out. And so what I would expect is that I could have big A going with big B. I could have one big A here, right? It's only one copy of a chromosome with a big A. And I'd have, you know what, let me zoom in. I'd have one copy here with a big A. I'd have another chromosome entirely with a big B. Or I could get a big A, moving in with a chromosome that has a little b, right? This one to this one, and then that one to that one. So those are 2 options there. What's the other option for the other chromosome? So here we have one possible zygote with big A, big B, another zygote with big A, little b. And of course, so far we have big A, big B, or big A, little b. Now we can also have to consider little a, big B, and little a, little b. So now we have a possibility of having a little a combined with a big B and a little a combined with a little b. So we have 4 different kinds of zygotes that can be produced. 4 different kinds of zygotes that can be produced because the genes aren't linked. They're on 2 different chromosomes. The one gene is on chromosome number 1, while the other gene's on chromosome number 2. And so after meiosis, you can have 4 different zygotes. Same thing over here if you look at, but of course in this parent, you'd only have little a. You still only have little a, little b, because that's all the only options this person has to give is little a and little b. So this person over here, really, if you think about it, this person over here only has really one kind of gamete it can make, or she can make, or he can make. It's only going to be either all of their gametes, all of their eggs, all of their sperm are going to be little a, little b. Why? Because those are the only 2 they have, whether they're linked or not. So that's going to be important when we start to think about what this all means as far as answering these questions. Okay, so now let's go back and look at the question. List the possible genotypes of the offspring if genes big A and big B and separate chromosomes. So think about the genotype when you're looking at if they were on different chromosomes, and I drew it out here. Now I think you guys could have handled actually putting them on different chromosomes and seeing what kind of gametes they would produce. All right, now here they say if the female Drosophila had been homozygous for the dominant wild types, there's a homozygous for these, so they would look like this, right? CU, CU. Now is CU capital C, capital U, or capital C, little u, capital C, little u, and big E, little b, big E, little b. This is actually one variable, by the way. So don't think that they're using a 2-letter system. where we would use big A, little b, etc. This is one dominant allele. This is another dominant allele. Here's another dominant allele. Here's another dominant allele. These two are the same genes, and etc. So you have this dominant trait, and they're asking you if the female Drosophila had been homologous or dominant type, wild type alleles. The genotypes of the F1 would be what? So if the female, which would be from the female, so if this female, instead of having these recessive traits, right, little c, little e, had big C, big E, then what would that do to the possible, what would that do to the genotypes of the F1? This is F1, right, the first group. Well, as it stands, the genotypes would go from having heterozygous, right? Why do I say heterozygous? Look at it carefully. Here's the dominant, here's the recessive, here's the dominant, here's the recessive. So heterozygous, right? Same thing as if it were big A, little b, big A, little b. But instead of big A, little b, they have big CU and little cu and big EB and little EB. All right. So again, this is also heterozygous, same. Because you have one dominant here and one recessive here. Now these are linked. And why do I know they're linked? If you look at the drawing above, you can see big C, little u, big C, little u. They're on different chromosomes here. Big E, little b, and little e, little b. Two different chromosomes. These two are on one chromosome, and these two are on another chromosome, so they move together. These two move together when the genes are being passed around during meiosis. Same thing here, right? Okay, so here there are two heterozygotes. So right now, and in this situation, and because this is recessive for both genes, when this female gives these genes over, the male only had the recessive of both genes. The F1 generation has 50% heterozygote and 50% homozygous recessive, right, for both genes. Now, what are the genotypes for the F1 generation if instead of having little c, little e, right, you had big C, big E? So in other words, both of these groups were dominant. The F1 generation would be what? All of them would be big C, little u, little c. Let me do a different color just so it's visible. Little c, little u, big E, little b, and little e, little b, right? The recessive, they're heterozygous. Why? Because this would be big C, big E, and then you'd have basically a copy of what's happening here, happening over here. And what would the phenotypes be? Well, they'd all be the same. They'd all look like these two, right? They'd all look like these two. So they would all look like this fly, these flies. What does that mean? That means straight wing, gray body. So they're all straight wing, gray body. So how does linkage decrease the amount of genetic variation you can have? It requires the two genes to move together. You can't, it's very difficult. It restricts the movement, the recombination of genes. the closer they are together, the more they're linked, the closer they are together, the less likely there's going to be crossing over between them as well. But linked genes, when they're on the same chromosome, they're going to move together, which means you can't get, it's more difficult to get heterozygotes between two genes, big A, little, I should say, no, I'm sorry. It's going to be harder to get a bunch of different combinations of two different genes in a gamete if they're on the same chromosome. If they're on different chromosomes, it's easier to recombine them, to have different combinations, as you can see here. All right, let's move on. So what is recombination? Now this is what's happening. So again, I'm sorry for the text message showing up there. What is genetic recombination crossing over? So with genetic recombination, instead of depending on whether the genes are linked or not, and again, the closer you are together, the more linked they are, and therefore the less crossing over is going to happen between them. As they cross over, you'll see that they produce, see they cross over here, you see they recombine, they give you the same kind of variation as if they had been on two different genes. Alright, so A and B, big B are linked, little a, little b are linked. But as you cross over, there's an exchange of DNA increasing the genetic variation. If they didn't cross over, if they hadn't recombined, then the linked genes are stuck with themselves. And if you look at these two are not recombined. In other words, the parental gene combination, big A, big B, the linked genes are still linked. Big A, big B, little a, little b, and little a, little b, little a, little b. So this is not something that you see the one parent had little a, little b, and that same parent had big A, big B. So if you see this gene combination down here, you're not surprised because, well, that's what you had, and it just so happened that this zygote got this combination of chromosomes, and this zygote got this combination of chromosomes. It's not a surprise, the parent here. Parent 2 can only give the recessive traits, so this is the only chromosome that parent can give. But these two show evidence that something's going on. This is evidence that there's been some recombination, some crossing over. All right, so describe the effects of recombination on the inheritance of genes. What it gives back, it gives linked genes the ability to have that variation that was missing because they were linked. Explain how recombination increases the amount of genetic variation in offspring. Well, I hope you understand that because you have these two different kinds of combinations, because of crossing over, you have different possibilities that you didn't have before you crossed over. Again, I'll refer you to the video from class where we went over this. And look at these combinations here. You can look at all the different phenotypic changes. Gray and straight winged gray body, curled wing ebony body, straight winged ebony body, and curled wing gray body. When you looked at before crossing over, the only two options you had were straight wing gray body, curled wing ebony body. Those were the only two options you had. Now in the end, you have 4 different kinds of flies from the same two parents. And why is that? Because you were able to recombine those genes. You're able to switch things up. All right, let's look at the next. All right, so what are the advantages of sex, and why is it that larger organisms need sex, right? So this is a really detailed example of what we've been talking about in class, and you can you can look and at these examples and look and see kind of this competitive idea of why why sex is necessary among larger organisms, and in fact the more complicated organisms. The key is that when we're talking about sex, we're talking about recombining DNA. You were talking about increasing genetic variation. When you increase genetic variation, you increase variability in a population. So therefore, you increase the odds that someone in this population is going to have the right combination of genes to survive for right now. Also allows for recessive genes to be kind of put away and not be impacted, and you having a recessive gene not impact your survivability, as long as you're heterozygote, of course, and or the conditions are appropriate. So explain how sexual reproduction confers an advantage over asexual reproduction. We just talked about that. We talked about it in class. Explain why species that produces asexually can outperform sexually reproducing organisms in some situations. Look at this question here and think about what these creatures had to do in order to survive and why. What kinds of situations would make sex a disadvantage, right? I'll give you a hint. In situations where you can't find the opposite sex. Again, take a look at this section here. So you describe a situation where species that reproduces asexually might not be favored. So think about it. Think about this. And then think about this. And then consider this question. Contrast sexual and asexual reproduction with respect to inheritance. That goes back to the idea that when you're talking about sexual reproduction, you're recombining, you're increasing the genetic variation. Where in asexual reproduction, you are gonna be whatever your parents were, whatever their parents were forever in a day. That's not going to change. Your genes are not going to change except for mutation. Asexual division doesn't allow for any kind of increased variation in increased genetic variation. Explain how reproductive strategy involving alternation of sexual and asexual phases might benefit a species. So think about why a species might go from being sexual, in other words, producing gametes, halving the DNA, recombining them, and then combining the gametes into a new organism, sexual reproduction, or asexual reproduction, switching from one to the other. Some plants do it. Look here at aphids and look at what they're doing. What's their advantage? Why would they do that? So I don't want to tell you the answer. This one we went over in class is just simply an example of a gene, so you can see how many letters there are in a gene. It's a very relatively small gene, right? So it's a bacteriophage. So it's a virus that attacks bacteria. So it's a tiny little thing. And you can go ahead and do some math and do your calculations. I don't think I need to help you with that. So this is the chromosome structure. I think it's interesting. Again, the chromatids are here again. I mean, you just keep getting this same process over and over. In chromatid, centromere, chromatin fibers, over and over again. You see the same structures. Nucleosomes, what are they? This is one of the vocabulary words. Nucleosomes are this kind of group of histones, right? So nucleosomes consists of DNA wrapped around a protein core. The protein core are histones. So the nucleosome is the DNA wrapped around the histones. That's a nucleosome. It's just that simple. And then you have the DNA molecule, which is double-stranded, held together by hydrogen bonds. All right. These banding patterns are there because of I don't think there's much to discuss here. I think you should be able to handle this. You take a look at. There's some reading here you should do. All right. These definitions should be able. You should be able to tell me why they're significant. Why each of these are significant. What is chromatin? What's DNA? What's a histone? What's centromere? What's a chromatid? You should easily be able to write that out. Each human cell has about 1 meter length of DNA in the nucleus. Does discuss the mechanism by which DNA is packaged into the nucleus and organized in such a way that it does not get ripped apart during cell division. I would refer you back to the previous page on the chromosome structure. I'll give you some hints. The protein histones, the DNA. Why? Remember, what are we talking about packaging? When it says packaged, we're talking about packaging, we're talking about condensing them. What does that mean? How are the two chromatids held together? What are the proteins involved? So look at the previous page and think about those structures and think about how you're doing it. Think about braiding hair. Think about a rope. When you braid rope, why is rope braided just like hair is braided? Because it makes it stronger. Why does it make it stronger? All right, let's move on. Karyotypes, this is just, again, the banding patterns of each of the individual chromosomes. Chromosome 1, banding pattern. Chromosome 2, banding pattern, etc. There it is. And, of course, you have an X chromosome and a Y chromosome here. Different banding patterns, again. Different shapes, different places where they're attached to different centromeres. Here's these beautiful scanning electron micrograph of chromosomes. I mean, they're absolutely gorgeous. You can see that this is why we drew them like this, because that's what they look like. And, of course, here's the X and Y. You see, that's why we draw this so small, because it is so much smaller than the X. The X has many more genes on it. That's why a male has an X. The female can deal without having a Y because the genes on the Y are not essential to life. You can survive without the Y genes. But there are so many genes on the X, you need a number of them are very important to life, and so you need it. By the way, here's some interesting organisms with 2N, and they're 2N, they're all diploid. Here's their 2N numbers, they're pairs, right? So 46 has 23 pairs, and 48, so how many pairs is 48? Obviously 48, 24 pairs, right? So this is 23 pairs, this is 24 pairs. Etc., all the way down to the goldfish having 94. So you can see the number of chromosomes doesn't necessarily mean an increased complexity, since a goldfish, I would argue, is simpler than a human, and in both body size and complexity. Goldfish has 94 chromosomes, where human has 46. So the number of chromosomes does not equate directly to how complex the organism is, which is not necessarily intuitive. You'd think the more chromosomes you have, the more DNA you have, the more DNA you have, the more complicated the organism is, but it doesn't seem to be true. So invertebrates, as you see here, in a honeybee can have 32 or 16. See, they can actually change how many, how strange that is. Most species, we keep our chromosome number the same, but some change it up. Oranges change it. Notice there's no corn, because corn is crazy. But oranges have 3 different 2N numbers, 18, 27, or 36. Interesting. All right. So moving forward, what's the difference between autosomes and sex chromosomes? Autosomes are this body cells. What's the difference between these and these two? Look at them. all of these are something, these two are not that thing. That's what you want to explain there. Why are these two chromosomes different from all the other pairs? This is a pair here, this is a pair here, this is a pair here, this is a pair here. Why is this pair so different from all the other pairs on this chart? The karyotype tells you what? It tells you how many chromosomes, how many copies of each chromosome you have. You can look at them. 321s, you have trisomy 21. That's also known as Down syndrome. So you can tell whether you have some chromosomal abnormalities or not, what aneuploidies you have. So here's what they would look like in a micro. This is actually the lab you would do. You'd prepare the karyotype by taking it and putting it in a test tube. You'd separate out the components. You'd take the precipitate out and you put it on the slide. You spread it out on the slide, and there you have it. There's these beautiful little things that are called chromosomes. And then now that you have that chromosome and you can, it's been condensed, you fixed it, now you can start organizing them. These two look alike, this one, and you can start separating the homologs. So this is a homologous pair, right? Here's, and so this is 44 plus an X and a Y. So 44 and X and Y is what? 44 plus 2 is 46. So 46 or 23 pairs, the X and the Y being one of the pairs. There's the X and the Y. And there's a female. Notice instead of an X and a Y, she has a what? 2 X's. Notice the male's Y is much smaller than the X. All the others are the same size. It's the X and the Y that are different. Here's 2 X's and here's an X and a Y. So you can write that down, write down the number. You should put this on the blank page that I printed, because remember I printed 2 blank pages. I really meant for one of the blank pages to be behind this one, but I messed that up. So you're going to have to take these answers and write them on the back of one of the blank pages. So this is what you have to cut out, right? And if this is on the back of the page we just looked at, so when you cut these out, you're going to mess up the other sheet that we just passed by. So that's why you have to write your answers on the blank sheet. So you're going to cut these out, and then you're going to organize them to, here's the blank sheets, the 2 of them. You're going to put both chromosomes number 1 here, both chromosomes number 2 here, etc., all the way down the line, and then one sex chromosome here and one other sex chromosome here. So you're going to choose that, you're going to cut them out, you're going to paste or tape them onto these little spaces, and you're going to get that done. And how are you going to tell which chromosome's which? Look at the chart that we just passed up. Rewind and look at it. You need to know all these vocabulary words. By the way, bivalent is the same as tetrad, right? Tetrad and bivalent are the same. They're 2 different ways of saying the same idea. The rest you can look them up and make sure you match. Here's a really great case study which looks at genes, and you can take a look, and after reading it, go ahead and answer these questions. All right, variation. Again, here's some variations. Here's a lighter colored albino zebra. Here's a zebra. Here is some variation in color on the skin. Here's some variation in color and size of the corn. Again, there's color skin variations, color skin variation, sizes and shapes and hair textures, etc. Here's in chickens, the combs, what we call the combs. These are roosters. All 4 of these are roosters. These have a pea comb. This has a rose comb. This is a walnut comb. And here's a single comb. And they're all chickens. They're all the same species, but there's variation within that species. Okay, so we have qualitative traits, and we have... So when we're talking about discontinuous variation versus continuous variation, discontinuous variation is variation that's not changing gradually, but it's either one or the other, or in this case, A, B, or O, or A, B, or O, right? So it's one of 4 different blood types, right? Where something like skin color variation is going to be slightly different all across the area. So you're not going to have a black or white. That would be discontinuous. A continuous change would be a little darker, a little darker, a little darker, a little darker, a little. So when you have a gradient of change, then you have a continuous variation. In other words, you have the variation, the differences just keep changing over time. This is discontinuous here. You have either this striping or you have albino, right? This is also discontinuous variation, right? So when you have continuous variation, you're going to have a kind of a gradation of changes from one color from one end to the other end and somewhere all in between. Where you have discontinuous variation, you have one or the other or the other or the other. There's not this just large change of different types, right? So skin color is a good example. The height of humans, right? So you can have very short humans, you have very tall humans, and you have the kind of the average height of humans, right? So sources of genetic variation are dominant alleles, recessive alleles, mutation, crossing over. Remember, we talked about that about 1,000,000 times. Independent assortment, gene interactions are all part of this increase genetic variation. Of course, the environment also is going to increase or decrease variation. So how much water there is available, how much food there's available. In the case of trees, the soil type, acidity. This is going to impact the phenotype, and so you're going to see different kinds of phenotypes. Genetic variation. This is the source. Phenotypic variation, in other words, small or large, green or purple, rough or smooth. What does it look like? First is impacted by genotype, which is impacted then by these factors, but then is also impacted by the environment. So you might have the genes to be tall, but if you don't get enough nutrition when you're young, those genes never never allow you to grow, or that lack of food doesn't allow you to grow as tall as your genes might have allowed. So let's take a look at the environment and its impact on phenotype, right? You go ahead and take a look at it, read it, and then giving appropriate examples, distinguish between genotype and phenotype. You should be able to do that. Identify which of the following phenotypic traits is continuous, quantitative, and discontinuous qualitative, right? Wool production in sheep, is that continuous or discontinuous? Again, continuous is quantitative. So that means there are numbers that you can associate with it so that you can get averages and highs and lows. Discontinuous tends to be qualitative, rough or smooth, or shape, a specific shape of one kind or another. All right, so when we're looking at body weight of mice, that's quantitative, right? That's something you could put a number to. Quantity, right? There's a quantity or quantitative. It means you're going to put a number to it. So that's going to be continuous, right? There's going to be some kind of distribution that you can look at. So you can continue reading these and answering the questions. You can look up what Klein is. What do they mean by that? It says it right here, but you go ahead and look it up if you need to. All right, so I don't think that's too difficult. Moving on. Alleles, here again, looking at alleles, the different kinds of alleles, what's heterozygous, what's homozygous dominant, what's homozygous recessive. You should be able to say that, to understand that. based on the reading, based on what we covered in class, I mean, I don't know what else to say. This should be pretty straightforward. Here's Mendel's experiments, and you should be able to work this out. I don't need to read this for you. What would I suggest you do? You should be able to figure, I think, just about everything on this page out. This is what he did. This is either round or wrinkled. That's obviously, hopefully you know that round or wrinkled are phenotypes. And he didn't know anything about genotypes. He was just counting phenotypes. And he was coming up with ratios. So 296 to 1, or wrinkled versus round, right? Wrinkled 1,850 to round 5,474 for this total. How do you get 296 to 1? How do you get 296 to 1? That's a question for you guys to figure out. How do you get a ratio based on these numbers? We'll go over it in class on Thursday. So Mendel's laws of inheritance, he came up with these observations, and laws are just basically what we see. We call them Mendel's law because Mendel was one of the first people to really describe it, but a law is just a pattern that we see in nature. And so here you see parents, generation, oocyte, there's meiosis, you see it happening, there's the eggs. So the theory of particulate inheritance is this idea that the characteristics can be passed on from one generation to the other in discrete units. So dominant, recessive. These things is particulate inheritance. He didn't know anything about DNA, obviously. So what he's talking about here, today we would call these genes, right? But then he had no way of explaining it because he didn't have this knowledge of DNA that you have. So law of segregation diagram on the right illustrates how meiosis and the two members of any pair of alleles segregate unchanged by passing into different gametes. The allele, the gametes passed on to the offspring. So how are they? They are separated and passed on. Law of segregation. That law of independent assortment says that in the diagram on the right illustrates how genes are carried on a chromosome. There are two genes, big A, big B, and they code for different traits. Each of these genes is represented twice, one copy allele on each gene of two homolog chromosomes. The genes A, big B, are located on different chromosomes, and because of this, will be inherited independently of each other. Independent assortment. If they were on the same chromosome, we'd call them linked, right? And they would move together. But because they're on different chromosomes, big A, and little b here, because they're on two different chromosomes, they can move separately. So you can get big A, big B, little b, big A, little b, little a, little b, big B, and little a, big B. This possible combination of alleles is possible because you have because the genes are not linked. That's number one, and they sort out into the gametes independent of each other. Basic genetic crosses, again, this is something you should be able to do. There's a Punnett square. We did it in class. You'll have a quiz when I get there on Thursday going over this. Obviously, as I said earlier, we're gonna have to move back all those assignments, their due dates. So back by I guess everything will have to be due on Monday, since Friday we're on a field trip. So what is true breeding? Think about it, understand it. All right, so true breeding means they're homozygous, right? You can do the reading. You should have done the reading in the chapter. You should be able to answer these. You should be able to tell me what are the different genotypic ratios here. If you have these two heterozygotes forming these four different kinds of gametes, what are the genotypic and phenotypic ratios? We did this in class literally today or yesterday. What's a test cross and how does it help? Okay, so let's think about this. You have a test cross. You have this thing that, let's just go ahead and use dwarfism again. Here's dwarf, and the person's dwarf with black hair. What does that mean? That person has an A something. We know we know A is dominant, so we don't know what is this second gene. We don't know unless we do DNA, some kind of molecular work, and actually sequence the gene. We won't know what allele they have there. And if they have black hair, then we know they have a big B, but we don't know, again, what's the second gene? What's the second allele? Is it big B, or is it big B, little b? We can't tell, because as long as it's the dominant trait, we can't tell if they have the recessive gene. So sticking to a simple Punnett square rather than a dihybrid cross. So sticking to 1, how do we know if this is homozygous dominant or if it's recessive? If we have the recessive trait, then life is good, right? So if we say the person's average height for this gene, we know that the person's little a, little a. Why do we know that? Because had they had a big A, they would be a dwarf, right? In this type of inheritance of dwarfism. So homozygous recessive is already easy to know. We don't need to test for homozygous recessive. We'll see it for sure, and we'll know. So the recessive trait is the only trait that gives us the genotype. So if we see the recessive phenotype, we know what the genotype is immediately, because it can't have the dominant version of the gene, otherwise it'd have the dominant phenotype. So in this case, let's look at dwarfism. How do we tell if this gene, right, if this allele is dominant or is it recessive? Are they heterozygote or are they homozygous dominant? We don't know until we do a test cross. So with a test cross, we're going to cross big A, whatever they are, with a creature who's recessive for that particular trait. So let's go ahead and say that the cross we're going to do is with an average height individual organism. So there's two possibilities. Either this second one, and I'll do it in blue here. This second one is either an A, or this second one is an or here. Or this second one is a B. I mean, so not A. A capital A here, right? Dominant, or it's a recessive version of the gene, and it's a lowercase a. So if we took an average height person, which would have the genotype little a, because that's recessive in this case. For an average height person, and we cross them with this dwarf, and we made many babies. We had to make a lot of babies, so that's another story. So we made a lot of babies. Let's say it's not human. Let's say it's a type of dwarfism for insects. It's a dwarf insect, right? So, and the dwarfism is inherited by the dominant trait. So if you do a test cross, the only way, let's take a look. Let's assume that the second letter is dominant, so that's homozygous dominant. 100% of the progeny, if it's homozygous dominant, 100% of the progeny with a test cross will be dwarfs. They'll all be dwarfs. There will be no average height beetle or insect or what have you. If, however, it's a heterozygote, then you're still going to get a bunch of dwarves, actually at least 50%, but then you'll get 50% average height. So you'll quickly see if this was the second space was filled by a recessive allele, because if you a test cross is when you cross an unknown genotypic individual with one with a recessive, homozygous recessive for that particular gene. Because then if you do get a recessive individual, if you in this case an average height individual, then you know that that second letter was indeed a lowercase or a recessive gene. Because had it not been recessive, there is no way that you could have recessive zygote, a recessive phenotype. And that's all explained here. You should be able to answer the questions pretty straightforwardly. So here's some more crosses and some more basically Punnett squares. Here they're asking you to fill in, the gametes, right? How many different kinds of gametes can this produce? A big B here and a little b there. That's pretty much all they can do. Here, big B and little b, and then we produce those two different kinds of gametes here, two different kinds of gametes here. Combine them, and you're going to put their genotypically combined. Their combined genotypes here. So this is basically what you see going on up here. You're going to do for all four of these. Easy peasy. Now the genotypic frequency and the phenotypic frequency, we did that in class. You should be able to handle. Incomplete dominance and codominance we did in class yesterday. So again, you should be able to fill that out pretty quickly. Same thing goes with this section. By the way, when they say that this cattle here is a roan, they mean there's two different colors, right? There's a brown and there's a tan. That tells you something about the genotype, right? All right. So here's some more of these drawings. ABO blood group. It's very interesting. Here's some more of these crosses you're going to do. Instead of guinea pigs, you're going to have humans. You can do that. And here's some more guinea pigs and their genotype and phenotype and dihybrid crosses. And here you're going to actually do a dihybrid cross. So this is pretty much walking you through the answers one at a time. Dihybrid cross with linkage. You know what? You can get rid of this one. Just don't do it. It's not worth our time, and it's getting late. So go ahead and delete that one if you'd like. If you want to do it, you can meet me after school and we'll explain it. We'll go through it. Here is a really nice, interesting example of an allele that if you get it, you're dead. So it's interesting. So some alleles that really end up causing some real serious issues. And some are recessive, some are dominant. So take a look at that one. I think it's a pretty interesting one, personally. Here again is some more Mendelian problems, dihybrid crosses. You can figure it out. I'm sure you can do it. They're real problems, so that's really interesting. In other words, these are real genetic inheritance situations or cases. Again, more dihybrid crossings, and genotypes and phenotypes. What's going on? Incomplete dominance and codominance. Test crosses. Human genotypes, dominant, recessive. So some interesting phenotypes that you can check and see if what your phenotype is. Right. So if for instance, if you have if you're right-handed, right, you have R, capital R, you have the dominant form of the allele. If you're left-handed, you have the recessive form of that allele. Our left thumb on top, etc. And you can go ahead and look at each of these are characteristics we all have. So here's your traits, and you can fill them out, and you can try to figure out your genotype based on your phenotype. Follow the directions, they're step by step. whether you're male or female, the sex determinant, you should be able to figure that out, especially with humans. A little more complicated with other organisms, but you can go ahead and take a look at that and read. Take a look at all the details that you need to look at there. Genotypic imprinting. Oh gosh, really, I'm kind of pushing it on this one. Almost not even worthwhile looking at it, except for because of its interesting nature. So there's more than just genes. It's more than just a sequence. It's this idea that there's a whole nother level to information in the DNA, and it's not just the sequence of the nucleotides, but then how methylated they are, and we don't need to go into details what methylated means, but how that DNA molecule has been changed by adding what's called methyl groups to it. And here's another one, sex linkage, and that we'll have to talk about in class. But I would still do the problems, and you'd see that's going to get you ready to really take advantage of those discussions. So inheritance patterns, you can take a look at them and see how genes are inherited. Here's a pedigree chart. I'd like you to draw it before we draw it in class. I want you to give it a try. The directions are there. The examples are there. I want you to draw them so that when we get to in class and we draw them together, it's a productive discussion. And here's reading the pedigree charts. Take a look at them. There's a key. There are some keys that you need to know, and so get used to reading these. Some more pedigree charts. Here's genetic counseling, which is an interesting idea and topic. By the way, here's chromosome number 21, and it's on the wall too, but here's chromosome 21. There's 3 copies. You have 3 copies of chromosome 21, and you have something called Down syndrome. Polygeny, or epistasis, and pleiotropy. These are interesting concepts. I think I have to wait till next week, but give it a shot to get it done. Complementary genes, also something we should try to hit on next week, but get a shot and look at it. Polygenes, again, each one is broken down. And by the way, remember we said that earlier that skin color is one of those continuous, right? So you can give skin color a number. You take that skin color, real light skin, and give it a 0. This is a very pale skin. Or you can go to what people, again, I don't like the very pale and then to black, because there's no one that's, I don't know. It's just going from 0 to 6 is a number system, which I think is more productive when you're trying to describe continuous versus discontinuous. So going from 0 to 6, we give it a quantity, or a number. Once you have a number associated with it, you can see more clearly why it's continuous. Remember I said it's like a gradation, just like we talked about concentration gradients. We talked about when you in your art class, when you talk about doing pencil shading gradients, you can go from lightest to darkest and everything in between. And because of that, we call this a continuous variation. All right. So you can see that there's more than one gene involved in this. That's why we call them polygenes. So what color your child turns out depends not only on your genes, but on your spouse's genes. And it's not just one gene, it's several genes. So again, back to the idea of discontinuous versus continuous. I think it's worthwhile to have that discussion. Excuse me. You go ahead and you can do this lab in class today if you have a chance, if there's time. Start collecting some data as far as tall and short to fill into those charts. Epistasis is this idea that one gene impacts the other gene. we'll talk more about that next week as well, maybe tomorrow. And here you have, again, some more examples to look through. I love this. I love that they use dogs and cats. As you know, I have both, and some really interesting, combinations and examples here of all these things that we've been talking about. And there's genetic variation and then phenotypic variation within different groups. Look at feline, the different colorings of felines. They're all of these are the same species. They all can produce progeny, but what you'll see is that each one of these has very different shadings, right? Very different colorings. Here's what genotype is that cat? Has that cat, right? So you can go through and do collect some data and make some decisions. Of course, there's your crossword puzzle. I don't need to explain how to do that, I don't think. And that's it. And we're done. So I hope that you're getting your work done. I hope that you're getting these key terms. Some of the more advanced stuff, we might have to leave some of it behind just because we're behind the schedule, as I've mentioned a couple times. I would love to discuss polygenes with you and epistasis and some of the other concepts. But we're going to have to get moving. We'll make a decision together on Thursday. All right. Again, sorry I couldn't be there with you today. Have a great day.